My name is Robin Shaw, and I work primarily with Wilbur Lab. Unfortunately, they're unable to come because now they're at the NIH. My mentor is Stephen G, and my biology sponsor is Dr. Victoria Bouch. So YAP-associated protein, also known as YAP, is a modular adaptive protein with multiple protein-protein interaction domains, and it can function as a transcriptional coactivator as well as a scaffolding protein. Now, when YAP is absent in mice, we see failure of embryonic development after embryonic day 8.5. Specifically, we see defects in yolk sac vasculogenesis, chorioallantoic fusion, and axis elongation. In addition, recent studies have shown that YAP expression is increased in cancer cells. For example, here, overexpression of YAP in a YAP homologue in Drosophila causes enlargement of the liver. Thus, studying the regulation of YAP could provide insight onto agents that are causing developmental defects and cancer. Now, every gene in a cell is expressed by processes such as transcription and translation, by which the information in the DNA is carried by the RNA into the cytoplasm and translated into protein. However, not every gene is required for every cell. Thus, gene regulation is very important. Gene regulation can occur at different levels during this process, and primarily the main processes that gene regulation occurs are transcription, post-transcriptional, and translational levels. I specifically focus on the transcriptional level. So in transcription regulation, a promoter is required to regulate the expression of the gene at the RNA level. And traditionally, a title box is known to recruit RNA polymerase II to initiate transcription of a gene. And this can be accompanied by a cat box as well. However, not every gene has a title box, and studies have shown that there are elements of a titleist promoter, such as a CPG island, an initiator, and a downstream core promoter element that can actually also recruit RNA polymerase II to the promoter. In CPG islands, there are also GC boxes, which a transcription factor known as SP1 binds to. So YAP actually does not contain a traditional TADA or CAT box. So it suggests that it must contain one of the elements of a titleist promoter. When I did analysis of the promoter on an online database, I discovered that the region 150 base pairs upstream of the transcriptional start site is highly conserved across species. And thus, this is what I've been focusing on. So in order to study the different regions of the promoter, I amplified different parts of the promoter and directionally cloned them into a gene expression vector which has a luciferase gene. And I specifically cloned these into the vector using specific restriction enzymes. And the gene is located in front of the luciferase gene. And this is very important because each deletion construct serves as a promoter, or it simulates as a promoter for the luciferase gene within the cell. And I designed constructs ranging from about 3,000 base pairs to about 150 base pairs. And these numbers are relative to the transcriptional start site. So I transfected each vector containing the deletion construct in NIH323 and M1 cells. NIH323 cells are mouse fibroblast cells, and M1 cells are mouse kidney epithelial cells. And during the transfection, I complexed the DNA with the lipid, which enters a cell via endocytosis. And this complex, the DNA, uses the cellular machinery to produce the luciferase gene. So after 24 hours, I collect the cell lysate, which contains the luciferase gene, and I incubate it with the luciferase substrate and measure the activity to determine how much luciferase was produced by each deletion construct. So when I performed these transfections, I observed that the minus 141 plus 28 construct maintained the highest level of promoter activity in both cell types. In addition, I also observed that the larger deletion constructs had lower levels of promoter activity, and this could be due to repressive activity, which would require further study. So once I've narrowed my focus down to the region 150 base pairs upstream of the start site, I searched an online database that would identify potential transcription factor binding sites that are located within this region. And I actually found multiple putative SP1 binding sites that are located within this region. And I specifically chose 2 binding sites, which is about 35 and 45 base pairs upstream of the start site. And I mutated 2 nucleotides within a sequence that closely matched the consensus SP1 sequence. I observed that the first SP1 mutation, the first mutation in the deletion construct reduced promoter activity to 40% from the normal deletion construct in 3T3s and 60% in M1s. I also observed that the second mutation had no effect on promoter activity in the 3T3 cells. However, it actually doubled promoter activity in the M1s. And the best explanation for this is that the mutation I made created an activation site, allowing another transcription factor unique to M1s to bind to that promoter. So once I know that the first mutation was critical to promote, the first site that I mutated was critical to promoter activity, I wanted to confirm that SP1 specifically bound to this site. And I performed an electromobility shift assay. In this, I isolate nuclear extract from both cell types, and the nuclear extract contains the transcription factors required for initiation of transcription. I also designed a DNA oligonucleotide, which is labeled with an infrared dye that fluoresces at 700 nanometers. And this DNA nucleotide contains the region of the promoter that includes that putative SP1 binding site. So when I incubate the nuclear extract in the DNA oligo, I see a shift in the band of the oligo if a transcription factor binds to that region of the DNA. And that reduces the mobility of the complex under an electric current. And when I add an antibody, I add an antibody to identify the specific transcription factor that is binding to the DNA. And if there is binding, I see a supershift, which further reduces mobility, as you can see here. So when I did this with 3T3 and M1 nuclear extracts, I'll just go through this slide. And the first lane is just the DNA oligo with no shift. And the second is a positive control, in which I designed a consensus SP1 binding site sequence oligo. And I see a shift in the band to this position right here. And when I add the SP1 antibody, I see a reduction in the intensity of the band rather than a super shift. Then in these two lanes, I did the individual strands of the YAP promoter oligo to identify any non-specific binding. And when I anneal these oligos together and incubate it with nuclear extract, I see a shift in the band similar to the one seen in positive control. In addition, I see the effect of inhibition with the addition of the antibody. And a good explanation for this, is that a lot of other studies have used the same antibody, SP1 antibody, with this supershift assay and have seen inhibition. Although further studies would be needed to actually confirm that SP1 is binding to this DNA sequence. So based on the data that I collected, I can conclude that the -141 plus 28 maintained the highest level of promoter activity. In addition, the mutation that I made in the first putative SP1 binding site reduced promoter activity significantly. And the electromobility shift assay data that I collected on that first putative SP1 binding site supports my hypothesis that SP1 binds to the YAP to control its transcription. To further study SP1's role in regulating YAP transcription, I can perform an SP1 knockdown and overexpression and see whether it affects the expression of YAP. In addition, I can identify other elements controlling YAP transcription. For example, in the deletion constructs where I saw reduced levels of promoter activity. And also, in T cells, YAP is actually not present. Thus, it serves as a place where I can start to study repressive activity and to determine whether this repressive activity and misexpression of YAP has any effect on function of T cells. Finally, I'd like to acknowledge Stephen G., a graduate student in the Milgram Lab, who has guided me through this study, as well as Dr. Sherry Milgram, who is my advisor, and Dr. Victoria Bouch, who has sponsored me in this project. And I'd also like to acknowledge the Milgram Lab, the UNC Department of Cell and Developmental Biology, and the National Institutes of Health for all their support.
Are there questions for Robin? Robin.
So I have two, a specific one and a more general one.
Okay.
So the specific one is, did you make an oligo from that other putative SP1 binding site that when you mutated didn't affect the expression? And does that super shift, or does that shift in the MSA assay?
No, I did not. I did not make an oligo because making an oligo is very expensive, and well, the transfection served as preliminary data to do the electromobility shift assay. So
Okay.
since I did not see any significant reduction, I did not make an oligo.
Okay. So the general question is, I wonder if you could give us more examples of where YAP might be transcriptionally regulated. You said at the end that it's not expressed in T cells, right? Is it expressed in all kinds of other cells, or is its transcription regulation highly dynamic during development or different cell types?
YAP is actually present very early in development, and it is broadly expressed, but there are other cells there are some cells that it's more expressed than in general, and there's other cells that it's not expressed at all. And I'm pretty sure that both B cells and T cells don't have YAP present. But I'm not entirely sure. I know that there's other cell types where YAP is not present.
Robin, were there other potential regulatory elements in that 148 base pair region upstream of the transcription start site? Why do you think the activity is remaining when it goes down about half, but there's still significant transcription?
So
What's going on there?
there's actually, I actually identified 4 or 5 putative SP1 binding sites. I've only mutated 2. It's actually been very difficult to find these binding sites because the entire region is very GC rich, and to match a sequence that would potentially be a binding site was very difficult. However, I've thought about doing DNA footprinting or well, specifically DNA footprinting, which would help me better identify these sites.
Are there other questions? If not, thank you.